So we've got kind of one last little run of, also I'll bring your, I totally forgot in the 9 a.m. this morning, I'll bring your tests in on Wednesday. I just forgot I have them. And of course you can come by the office and look at any of your tests that you want to at any point. Are you thinking about turning on? Is it flashing? Short. And I've had too much coffee. Yeah, you just, it's in like some weird standby that it won't come out. I just have to turn it off and back on again. Or at least that's the only way I can get them to work. So, okie dokie. Let's try that again. So on Friday, we had talked about medical applications of kind of biotech, right? We talked about vaccines, doing kind of diagnostics, testing for viruses and antibodies, and nope, there we go. And all these different things we can do, right? We kind of talked about CRISPR and potentials for gene therapy, and even things kind of as, that have been around long, like making synthetic insulin, right? So we've got obviously loads and loads of medical applications, but biotech is also really, really important in agriculture, right? Kind of feeding the planet is an ongoing problem for lots of reasons, some of which have to do with how much food we're able to make, a lot of which has to do with politics, but making food we can talk about. So we'll talk a bit about transgenic crops, what they are, what they do, why we do it at all. And so one of the main things that gets done to crop plants, right? And this is kind of the result of genetics, right? Transgenics, inserting genes for different things. But we create herbicide or herbicide resistant crops. So glyphosate, it's a really common herbicide. It's what's in Roundup, right? Kills pretty much all plants, more or less, by inhibiting their ability to produce an enzyme that they need to live, especially broadleaf plants, things like soybeans. And so some crops like soybeans, alfalfa, things like that, have been engineered to be resistant to glyphosate, right? So we've inserted genes that make them herbicide resistant to glyphosate, and we call them Roundup ready crops. And it actually imparts kind of, well, a good deal of benefits, kind of large, kind of broader reaching benefits that you might think, right? So if I plant soybeans that are Roundup ready, they don't have to be weeded. If the crop does not have to be weeded, I can do what's called no-till planting, right? I can just put the seeds in the ground. I can use glyphosate to get rid of weedy species that I don't want growing within the crop, and I don't have to till the ground around them. This is important because tilling, which we do, for lots of crops, tends to kind of cause nutrients and things to be leached out of the soil. The soil kind of decreases in quality over time. You lose moisture, right? This whole kind of thing that happens from turning the soil up. So you don't have to do that. So you kind of have a more sustainable version of farming in this sense. You can use a single herbicide, right? Because glyphosate is really effective. So if you can plant a crop that's not susceptible to glyphosate, you can get away with just using a single herbicide as opposed to spraying like loads of different kinds of chemicals, right, on a crop. And glyphosate breaks down in the environment. So it's not something that's going to kind of build up and accumulate in soil, in plants and things over time, right? So these Roundup ready crops are really useful and actually kind of environmentally positive in a lot of ways. And so in the US, 90% of the soy we produce, and we produce a good amount of soy, is GM, is genetically modified to be herbicide resistant. Another thing that is kind of top of the list in a lot of common crops, and we're hitting soybeans and corn, which are basically two of our biggest products. We create what are called Bt crops. And so a Bt crop is a crop that is resistant to pests, right? Our two big agricultural problems are weeds, right? Things choking out the thing we want to grow, and pests that want to eat the thing we want to grow. So Bt crops have had a gene inserted into them that codes for a protein that makes them pest resistant. So the gene is from this bacteria, Bacillus thuringiensis. And so Bacillus thuringiensis produces this, it's called a Bt enzyme, because it's just the initials for the bacteria. And this enzyme, when it's consumed by an insect, is converted into Bt toxin, right? And you don't really need to worry too much about the enzyme and the toxin and what it's doing, but basically this gene codes for a protein that when the insect eats it, the insect's own proteins converted into something that then kills the insect. It's kind of bad mechanics on the insect's end. But either way, this gene's inserted into a lot of crops to make them resistant to pests. So Bt maize is our second most common genetically modified crop, right? So corn. And then of course you'll end up with stacked crops that people will use, right? So you can have a crop that is both resistant to pests and Roundup ready, right? So we have these kind of modifications for kind of like big agriculture sort of problems, right? And then we have the golden rice example, which we already mentioned. And so this is rice that's been genetically modified to produce beta carotene, right? To be distributed in places where food is scarce or it's difficult to get, appropriate nutrition. And it's interesting for two reasons. So one, it's a kind of the second one listed. This could not have been done by conventional breeding, right? This is the way they've modified golden rice. Basically, it could only have been done this way. There's no way we could have like inched rice into this. There's no precursors, direct precursors to beta carotene in rice at all. And so what they did to do this is they introduced an entire biochemical pathway into the rice. So multiple genes coding for all of the enzymes in a whole pathway to get from some kind of, run-of-the-mill organic chemical to this beta carotene vitamin A precursor. And kind of a nice turn at the end of all this, because frequently large research doesn't go this way. The labs that created this golden rice have made it available for free with absolutely no commercial entanglements to any kind of countries, nations, regions that want to have it, right? So it's just been distributed to anywhere that it would be beneficial. And this is just, you don't need to know all these enzymes or have these memorized in any way, shape, or form. It's just to kind of show you what they did, right? So we have the rice genome, which is good size, and they basically inserted 2 daffodil genes and 1 bacterial gene to create this entire pathway, right? So a synthase, a desaturase, and a cyclase that gets you from this geranyl diphosphate, I don't even know what this is or what it does, right? But takes it through this pathway, through these enzymes to create beta carotene, right? So that you end up with that kind of orangey colored rice. Right? All right. Of course, as with any of these sorts of things, we've got some kind of social issues, different ways this stuff gets portrayed in the press, different worries that people have about doing things like this to things that we might want to consume. And so it's been resisted in some areas just because people have questions, right? People are unsure, they haven't sat through this class and heard all this stuff. So, they've got questions. So there's always this idea that these GM crops might not be safe for human consumption. And there is absolutely no research that supports that concern whatsoever. And if you think about it, it makes sense, right? Everything we know about DNA, how DNA works, how it is moved or not moved, right, around in your cells. Every time you eat, you are eating the DNA of something. And that DNA and your DNA don't interact at all. That's not, it's just kind of not how it works, really, right? So there's no documentation of adverse effects on human health, but there are concerns. There are valid concerns people could have about doing this sort of thing. There are potential effects, right, that we don't want to happen. So if we do things to corn or to soybeans, right, they have native wild relatives, right? There's other plants around that are closely related to them. We've already talked about, and we'll kind of continue to talk about in chapter 18, how plants are weird, they'll hybridize, they'll shuffle genes around, they do funky things. So it's possible that we could alter the genes in crop plants and those genes could somehow or another be hybridized or passed into wild plants. And we don't necessarily want that to happen. This could lead, right? There's a potential for loss of biodiversity, right? As we start kind of modifying things, and if those things affect wild things, right? There's all these kinds of downstream effects of doing things like this that could be concerning. And again, with this idea of hybridizing and passing things on, right? There's also around all of these agricultural regions, non-native plants, weedy species, invasive species. And it's possible that these kind of potentially even detrimental plants could gain this resistance, right? We could end up with a invasive, right? What if kudzu became like resistant to everything? We'd just be overrun completely, right? So there's those kinds of concerns, which I think are kind of much more valid and more worrying than some of the other things we worry about with transgenic crops. And I think that's just going to be a thing going forward between the medical applications and especially kind of the medical and the agricultural applications. These are just going to be conversations that we people, scientists, politicians, like the whole lot of us are just going to kind of have to keep having, right? And sort out kind of what we're okay with and what we think should and should not be done. All right, which is the end of chapter 17. So we'll run through a couple of these. I'll just kind of like pull a couple. What is recombinant DNA? All the way back at the beginning of chapter 17. Yep, exactly. So it's a DNA molecule that has genes from 2 different sources, 2 different species, something like that. Let's see. So what about gel electrophoresis? What's a question I could ask? So with gel electrophoresis, right, was that thing where we had a piece of gel and a buffer and we put DNA in it and it sorts the DNA, right? If I use some restriction endonucleases to cut my DNA up, right? A gel will sort them by what? By what characteristic of the DNA? By size, it'll sort them, right? Little DNA fragments and big DNA fragments. What is a knockout mouse or a knockout organism? Absolutely, yep. A knockout mouse or other animal or whatever is one where we've turned off a gene, basically. So we can't make knockout plants because plants are weird. So we make what are called transgenic plants. Well, we make transgenic plants. And so how do we do that? How do we get around the fact that we can't just kind of make them take up genes the same way? Yep, we use bacterial transfer, right? There's bacteria that are able to insert genes into plants. So we just put the gene we want in the bacteria. Bacteria puts it in the plant for us. Then we culture the plant from there. Cool. Right, and then this is just kind of reminding you of these different applications. We just basically ran through a bunch of them. We won't do that again. We'll go ahead. Of course, these review questions are on Canvas for you to check out at your leisure to do. So let me switch the slides out for these folks. All right, so we're going into chapter 18, our last. Gotcha, Rowan. Full chapter of material. There we go. Boop. Boop. And so chapter 18 is on genomics. And basically we're going to tack in kind of a section or two of chapter 24 if we have time, that are just going to build on this. So chapter 18 is about genomics. So we've kind of been looking very fine scale, right? Moving single genes, making specific changes to genes, right? And doing kind of research at that level. And so there's also research that kind of happens at the whole genome level, which is called genomics, right? So it's integrating kind of classical molecular genetics, which is kind of the stuff we've been talking about, right? With biotechnology, which was also, so we're just kind of pulling everything together to study structure, function, and relatedness of genomes, right? An organism's entire kind of set of DNA, right? Where you can look at multiple individuals' entire genomes and compare one to the other, right? And then start to say things in kind of a more holistic or synthesized sort of sense. So we'll start with mapping genomes. We talked about this kind of briefly when we were talking about Mendelian traits, right? Kind of watching patterns of inheritance in groups of organisms and kind of seeing how traits behave and are passed on, right? When we talked about recombination and crossing over, right? You can kind of guesstimate basically how close or far apart genes are on a chromosome, depending on how much crossing over recombination happens. But now with all of these kind of new sequencing and restriction endonuclease, all these other kind of biotech methods, we can be a bit more precise in our genome mapping. So this means we've got two kinds of maps, right? So genetic maps are what we talked about earlier. So these are just kind of talking about the relative position, not like the exact spot on the chromosome where a gene is, but right, we can look at recombination frequency, right? That whole thing where like, I can look at a few traits, right? Traits A, B, and C, see how much crossing over happens between them and say that like gene A and gene C are very far apart. Gene C and gene B are very close together based on the recombination frequency. So, but it turns out, because nothing can just be simple or easy, crossing over is not as random as we'd like it to be, right? As sometimes crossing over will happen more in some areas of your genome than other, or it'll vary between individuals. So we can learn a lot by genetic mapping, right? Looking at patterns of inheritance, but it's not super precise, basically. So we have physical maps, right? And this is the precise position of a gene in the genome, right? You know that DNA is a long string of nucleotides, right? And so people will look at DNA and just number them, right? This chromosome has nucleotides 1 to like 3,000. And I know that gene B occurs at nucleotide 574, right? So this is making like a precise map of a genome, right? Both of these use genetic markers, right? Traits, characteristics, or genes, or specific pieces of sequence that we're interested in, right? And then we see how they vary between individuals, either through kind of more biotechnical methods or by looking at inheritance patterns. So we've talked about genetic maps, so let's talk a little bit about physical maps, and we're just going to kind of briefly list and describe a few methods of making these physical maps. So we've got restriction maps, which are going to use those restriction enzymes that we've already mentioned. We're going to make chromosome maps, and then sequence tag site maps or STS maps. And so when we do these physical maps, the distances are measured in base pairs, right? So in those kind of, right, if you have a double-stranded piece of DNA, you've got a long string of nucleotides, and every nucleotide has its like matching nucleotide on the other strand. So we just call those base pairs, right? And we're going to kind of measure distance through the genome, and we're going to measure it in kilobases or thousands of bases. Because genomes are huge, like hundreds of thousands of bases. So restriction maps, don't worry too much about the schematic, but restriction maps are specifically for small DNA molecules, so less than 50 kilobases, less than 50,000 bases. So these would be things like organelle genomes, so if you're looking at the genome of chloroplasts or mitochondria, and viral genomes, right? So viruses have pretty small genomes. So you use restriction maps for those, things that don't have really, really large amounts of DNA. Right. Oh, let me pop back. Right, and so the restriction maps, because you use restriction endonucleases, so it's this similar thing we saw when people were doing kind of transgenic experiments in the last chapter. Basically, without getting too far in the weeds, you chop your DNA up into kind of variable amounts of pieces. You look at kind of tagging those pieces back together. This is all done mostly in a computer. So computer kind of sorts everything out. You see these kind of patterns of cuts given your restriction endonucleases, and you're able to kind of infer a physical map from that. But again, this only works basically if you're working with small pieces of DNA. If you're working with like hundreds of thousands of base pairs, computationally, it just becomes too much. Chromosome maps is for larger amounts of DNA. So this is kind of mapping at the whole genome level, but at a lot lower resolution. So basically we can stain chromosomes, right? There are pieces of chromosomes that will consistently kind of take up dyes in a specific way. And so we can kind of create these banding patterns, right? So this is an example of chromosome 9 and chromosome 22. And they each have this region that will take up this dye at a specific place on the chromosome, right? And this banding pattern is important because occasionally, right, we talked about these instances where different chromosomes will kind of inadvertently, not inadvertently, but will swap chunks, which really shouldn't happen. And in this case, when chromosome 9, right, has kind of this translocation event with chromosome 22, this results in a specific kind of leukemia. And so if you see leukemia in a patient, you can use this kind of banding fluorescent dye to see if this is the kind of leukemia they have, right? Because instead of seeing two separate red and green dots, right, at different places in the genes, or in the chromosomes rather, you'll see them kind of occurring together on the same chromosomes, right? Which indicates this person has this specific sort of leukemia. So this is used in diagnostics, among other things. But it's just kind of a rough map, right? You're just kind of watching these two specific pieces of chromosome kind of move around in the genome. So it's not for like making very, very precise gene maps. All right, then we have sequence tagged site STS maps. And these are kind of somewhere in the middle of other things. So these are using kind of short, unique sequences that occur within genomes. We've kind of mentioned that this is a thing before, right? It's things we can target with endonucleases or tag them somehow. And so if we have these little short, unique sequences, we can make primers for them, and we can PCR and amplify these short sequences, right? And so we can then like amplify some certain region of the genome, we can fragment it. And then if we look at these markers that we've put in there, right? If they stay together on the same fragments, it's kind of the same logic we use for crossing over. So if you end up with fragments and the STS markers are still on the same fragment, then they must be close together. If you have fragments that, if you have STS markers that always end up on different fragments, they must be further apart, right? So it's kind of crossing over sort of logic, but instead of looking for crossing over, you're just looking for these little markers that you've tagged in these genomes. And this mostly, again, this is not a super high resolution option. It basically just gives you kind of a scaffold, right? It's that idea that like this gene's close to that gene and this gene's far from that gene, and you kind of logic puzzle it together, which happens a lot when we're trying to map whole genomes. They're just so big. It's gigabases of data in a lot of cases. And so when you're trying to sift through that at this like literally molecular level, it does just kind of take a lot of like, well, I'm going to break it apart and look at it and map it back together, and then break it apart and map it back together, kind of in this iterative process, right? To try and figure out where different things are. Right? And so we have our genetic maps that we get by analyzing patterns of inheritance. We've got our physical maps using all these different kind of DNA cloning sort of methods, watching how DNA behaves and figuring out where genes occur. And they are correlated, but they generally don't exactly match up for that reason that crossing over is not entirely random and it's not entirely consistent. So you may look at a piece of DNA and you may look at patterns of inheritance, and you may compare it to what you get when you do physical mapping, and probably you'll end up with things in the same order, but you may not have them in exactly the same place, right? Just kind of due to that slight variation in the way that inheritance will behave in different individuals, right? So they're comparable, but they generally will not line up exactly. But it's just that idea that genetic maps don't have as high a resolution as, right, as actual like molecular genomic physical mapping. All right, so we can map genomes. we can also, we don't even have to know the genes. We can also just sequence genomes, right? We've got tech for this. And this is a big kind of really growing really quickly kind of area of research or area of technology just in general. So we'll talk about a couple different sequencing methods. There's actually lots of sequencing methods, but we'll kind of talk a bit broadly about it. So the ultimate physical map, right? We've been talking about making physical maps by like coding things and making things light up and staring at things under microscopes. The most accurate physical map is just the base pair sequence of the whole thing, right? If I could just go through and read this molecule from end to end, that would be the most accurate physical map, right? And so we can do that. DNA sequencing is a relatively new technology. Well, I say that. It's come a long way in the last few decades anyway. So DNA sequencing is determining the actual sequence of nucleotides in the DNA, right? And all the ways basically that we have to do this involve us kind of mimicking DNA replication in an in vitro environment. So we're kind of back to that PCR idea, right? We're putting tubes in a machine, we're like flashing lasers at them while we watch DNA replication happen. And so a lot of advances in genomics are possible as this ability to sequence becomes faster and more affordable, right? Remember, we had that $3 billion human genome decade and a half long project, and we're down to what? Like $1,000 in 3 days for a genome. So things are getting a lot faster and a lot less expensive. And so the amount of data basically we're able to produce is growing exponentially. So Fred Sanger in the 70s developed what we now generally just call Sanger sequencing. There were similar methods out there, usually that involved larger amounts of radioactive materials. And so these kinds of sequencing are called dideoxyterminator sequencing. And a lot of people are really particular about you calling it dideoxyterminator sequencing, but nobody says that. We all just call it Sanger because nobody wants to say all that every time you need to order something from somebody, right? But they use dideoxynucleotide chain terminators. What that means. So you've got DNA. When DNA replicates, right, we've got one strand, we're making a copy, and we're tacking these nucleotides on as they match, right? And so one links on, and then we come in and we link the next one on, and we link the next one on. So what Sanger and some others figured out they could do is they could kind of make these nucleotides that instead of having a hydroxyl here, just had a hydrogen, right? And now this one, once this nucleotide attaches, when we're synthesizing DNA, the next nucleotide won't attach. We can kind of, this is why it's called terminator sequencing. So when this nucleotide attaches, there's nothing else will happen. We're kind of stuck here. And you can also code these, right? So say we've got adenine, thymine, cytosine, guanine. You can code them with fluorescent dyes, right? So I can kind of create, right? I can use polymerases and all these things, and I can make DNA start to synthesize itself. And so when one of my little terminator molecules gets on there, nothing else gets added, and then it fluoresces. So I know at this position is this nucleotide because it flashed red when I hit it with a laser, right? And then I run it again, and another nucleotide gets on there and stops kind of at the next spot, right? And then nothing else attaches on. I hit it with a laser. Oh, that one flashes yellow. It's a cytosine, right? And was initially an incredibly slow process because you literally did DNA like one nucleotide at a time like that. Right, so kind of like this, right? Right, this is the same thing as we saw in PCR. Right? We denature it, we heat it up. I'm like, okay, I'm going to sequence this piece. I'm going to cut it off. I'm going to PCR it and make loads and loads and loads of copies of it. I'm going to attach a primer. And then I've got, right, adenine, thymine, cytosine, and guanine little terminators. So I'm going to take these pieces and I'm going to run a reaction. This is like the most horrible, I cannot imagine, right? So I'm going to add these polymerase solutions. I'm going to start growing these strands. And every time one of these terminator hits, right, it's going to flash, right? It's going to fluoresce some color, right? And then it's not over. You denature the chains you grew. You run them out on electrophoresis gel, right? So I'm like, all right, my smallest one was only 4 nucleotides long. and it flashed, what did it flash? Yellow. So it's like, right. And they had to sit here and do this by hand for like years. It took years to make sequences. Insane, right? Eventually they got automated sequencers that, these single reactions that people were doing by hand for like ever and ever and ever. They got machines that could kind of do it, right? Computers that could make this output for you. And they got up to a whopping 1,000 bases a day by 87. We can do hundreds of thousands in an afternoon at this point. But so this is where we started, Sanger sequencing. And of course, all of these chemicals initially were like radioactive and horribly dangerous, and people were, everybody was full of tumors. All right, so it got automated. That was better, right? Because then I could put these in. This computer's like handling this every time something flashes, right? We hit it with a laser, we record these little, and this is cute. You still get these little graph outputs when you do sequencing. It's very satisfying, little rainbow color stuff. And so that actually is kind of still how, to that degree, it works. So we automated it, it got faster. And then we got next gen sequencing, or second gen, or whatever anybody wants to call it. Again, this one's got its more accurate name is massively parallel sequencing, right? So in Sanger, you're doing one sequence at a time, right? You're sequencing it out. In NGS, you are able to kind of basically put like thousands of DNA sequences on this little, it looks like a little microscope slide, this little plate, and kind of have all of these reactions occurring at the same time. And you have a computer kind of recording it all at the same time, right? And so this is just that thought, right? So we had this like $1 billion genome up until about 2004, right? Which is about the time we created NGS. And now we're down, right, to about $1,000 a genome. So the price dropped a lot, right? So how does next gen stuff work? So again, this is one of those things where there's lots of kinds of next generation sequencing, but they all kind of share some traits. So we won't go into each individual one. We'll just kind of broadly say how they work. So remember in that Sanger thing, like we denatured our DNA, we cut out the pieces we want. We made a bunch of clones of whatever piece we were going to sequence, made this what we call a library. So we don't have to do that with this, right? We're just going to take our DNA. So what I do, if I'm doing genomic sequencing, I take a sample from an organism, you do a bunch of, chemical bench stuff. You get this little tube with what we call just whole genomic DNA. I've just extracted all of the DNA that was in this piece of tissue that I collected. And I just send that tube off. They clean it up a little bit. And they put that DNA sample on what's called a flow cell. It looks like a little microscope slide with primers and things. And millions of these sequencing reactions, right? Kind of like what we saw in Sanger, but now it's like all automated and it's all happening at the same time, can happen simultaneously, right? And so it's all happening kind of in solution within this machine, and there's lasers shooting through there the whole time. So we don't have to do electrophoresis. We don't have to visualize this at all. It's all automated. It's all done. It's all basically done by a computer at this point, right? So we can sequence just millions of things all at a time, right? So that's cool. We can sequence loads of molecules at the same time, but we're technically producing kind of less information per molecule, right? Because we just kind of dumped it all on a cell and started, tacking little fluorescent dyes onto it, hitting it with lasers and stuff. And so the read length we get tends to be short, around 50 base pairs, right? Which is relative to a genome, not very long at all, maybe 1,000 base pairs. And so if you are wanting to sequence a whole genome, what happens, right? So if I send that DNA off and I want to sequence a whole genome, they run sequencer, blah, blah, blah. It runs for a day or two. Eventually it outputs this whole enormous file of what we call reads, little pieces of DNA, right? So they send me thousands and thousands of reads. And then I have to stitch them all together into a genome. This requires basically it requires supercomputers. So usually if people are doing this, they are attached to somebody's supercomputer somewhere. There are some sequencing methods that will produce longer reads. They just tend to be a bit more error prone, but So a lot of times people will kind of combine sequencing methods. Maybe they'll get kind of longer, less accurate reads and compare those to shorter, more accurate reads, and you start to scaffold things together. Right. So I've sent off tissues, not tissues, I've sent off DNA. I took tissues, I sent off DNA. I get back enormous files of reads. There was actually one project I did where you had to drive to the place with a hard drive. They couldn't even, there was no way to send it. You had to go like physically pick up your data. So how do you assemble this hot mess? Two ways. One is the clone config method. So this goes back to those mapping kind of methods we were talking about, right? So we kind of get these large clones, these large chunks of sequence, and kind of push them into continuous sequences by using that restriction mapping and STS analysis that we talked about in the last section, right? Then we take kind of these larger clones and then we fragment those into small pieces and then we sequence those. The trick is all of this is all great, like taking big chunks and chopping them up and mapping together and chopping them up and then sequencing. It's very circular because you actually already have to have a physical map for this to work. So this is if you're working basically with model organisms, right? If you're working with lab mice or Drosophila or nematodes or something we already have at least a version of a genome for, right? Because basically we're assuming that I have something to compare it to. So I can chop these things up, I can sequence them, and then compare them to something else and slowly kind of stitch all these reads together that I get from a sequencer into pieces and then get my version of the organism I sequenced, genome, right? But again, it kind of requires you already knowing what you're looking for. I work on non-model organisms, so I use shotgun sequencing. So shotgun sequencing, DNA is just randomly chopped up into bits. We clone it, we sequence it, and then we let computers spend days running algorithms, trying to figure out overlaps, trying to piece it all back together. Because for the organisms I work on, we do not have genetic or physical maps at all to compare to, right? So when we get sequencing data, it tends to look something like this, right? So I send off DNA, chop it up into a million little bits and just sequence it over and over and over and over and over and over and over again, right? And then some computer comes back in, right, and maps where things match and maps where things match. And eventually you get these larger, what we call contigs, contiguous pieces of DNA. Right? Eventually, you frequently can't actually put them together in order of an actual genome, but you can at least get long sequences of DNA based on these kind of overlapping sequences. It's tough. It's computationally tough. So you can use clone config if you have a reference genome, basically, or shotgun sequencing if you do not have a reference genome. All right. So let me check something really quick. Folks in the room, who has not done one of these already? It's an academic advisor. All right, 1, 2, 3. Nobody has. Okay. Oh,
Dr. Godwin, you're a robot right now.
Okay. Hold on. All right. So, folks on Zoom, Wednesday we're picking up at 18.3. Folks in the room, we have to do paperwork really quick. They did it last week. Let me do something that will take like 2 seconds.
Dr. Godwin, when you get a chance, I have a question. If you can hear me. I have a question, Dr. Godwin. Before I go, and I know you're passing out stuff for the in class, but the quiz that's due today, what chapters are those over? I cannot hear you.
How about now? Now?
Yeah, I can hear you.
All right. Stable microphone. Should be, I think, most of chapter 15 and then the first half of chapter 17. Does that sound right? Okay,
Thank you.
it's 15 and 17. I think we finished 15 on like Wednesday and then started 17. All right, so.
Perfect. Thank you.
Oh yeah, no worries. So this is to evaluate your academic advisor.
Great. How old is Linda? I'm right in chapter 24 now. There you go.
So it's front and back. The first part is just kind of numbered questions. This back has a few little short answer questions. It's completely anonymous. I'm not even going to take them downstairs. Subsequently, if someone doesn't mind taking these downstairs, that would be great. It's to room 110, office 110 downstairs. So. So yeah, if somebody doesn't mind running these downstairs. So I can't take these because they're supposed to be like anonymous. So once they're filled out, I'm not supposed to. So they just got to go in an envelope and down to like office 110 downstairs. Okay, thank you. Yep, just get everybody's in there and downstairs. Thank you. Eat. Oh. I don't know. Get it as close as you can. Hopefully they'll figure it out. I mean, oh cool, I left the recording that whole time.